across the UP fair, man. I breathe that freezing air, man. Of travel I've had my share, man. I've been everywhere.
Hi, this is Jim Islip with MSU Extension, and this is What's Up at UPREC on April 2nd, 2021. Today we'll have a look at the Winter Barley Hardiness project.
Hello everybody, today we're out here checking out our Winter Barley Hardiness study supported by the Michigan Craft Beverage Council. So the objective of this project is to look at winter barley hardiness to understand the potential of winter barley as an adaptation tool for climate change in Michigan. So why winter barley? Well, there's two kind of growth habits for barley, winter barley and spring barley. Traditionally for craft beverage production, for beer, the industry has favored spring barley varieties, but there's a couple of reasons why we think winter barley might have potential in Michigan. For one thing, it tends to yield quite a bit more than spring barley. Secondarily, it has some other advantages. For example, because of its growth cycle, being planted in the fall and harvested a little bit earlier the following summer, it can also potentially avoid some disease pressure, particularly Fusarium head blight and the DON mycotoxin that comes along with that disease. Another thing that's more relevant downstate is the opportunity for double cropping. So some of our colleagues at Kellogg Biological Station have looked at double cropping soybeans after winter barley, and because it is harvested a little bit earlier than spring, that's another advantage. So despite all those good things, there's a significant disadvantage, and that is winter hardiness. Winter barley is not very hardy. Compared to other winter cereals like wheat or rye, it tends to see winter injury or winter kill that kind of disrupts the whole system. So there's a lot of interest in understanding how winter hardiness functions in barley, what the important factors are that control the plant's response to environmental conditions, and what kind of thresholds in environmental conditions exist out there. Also looking at geographically, based on that kind of understanding, where in Michigan should we be producing winter and spring barley. So right now there's this sort of general split in the industry that we grow spring barley in northern Michigan and the Upper Peninsula, and we can grow winter barley in southern lower Michigan. But nobody can tell you today where that line is, perhaps latitude-wise, where we would advise growing spring versus winter, or where there may be some overlap. We think about winter barley then as possibly a tool for adapting to climate change because of the flexibility that it might have for some of the reasons that I mentioned earlier, but also because as things warm up in Michigan, we're expecting that that front, if you will, of winter barley production to move northward and to have more opportunities for winter barley production in other places in Michigan than we've had in the past or where we maybe have today. This project is funded by the Michigan Craft Beverage Council, and we have a number of collaborators on this grant. Brooke Wilkie from Kellogg Biological Station, Dean Boss down there at KBS. We also have a collaborator on campus. Emily Merowitz-Holm is a grass physiologist who's helping us with some of the laboratory portion, as well as Nikhil Jaikumar, who is a collaborator from Illinois. He's currently at Illinois State University. So this is a nice regional collaboration, and we have partnerships across Michigan. We wanted to have locations in a lot of different environments in Michigan. So we set out with the goal of having one of these plots in every hardiness zone in the state. And if you look at a USDA hardiness zone from Michigan, we have 6 hardiness zones, and we ended up with plots in 5 of those. So we did not get the coldest hardiness zone over in the far western UP, but we do have all the other hardiness zones, the warmer ones with more potential for winter barley, represented in this project. So we're in Chatham today at UPREC, and this is our location here. The next location would be Brimley, Michigan. And then we also have Empire down near Traverse City in northwest lower Michigan. We have KBS at Hickory Corners, and then we have Stockbridge, Michigan near Lansing as the 5 locations where this project is currently replicated. 4A would be our coldest zone, and that's the one we don't have. So we have 4B, 5A, and so forth heading south into the warmer areas. So as I said, this project is focused on winter hardiness. We have an issue where winter barley can be sown in the fall And then we come back in the spring and some of it has either been injured or actually killed by conditions over the winter and into the spring. And we end up with no crop or a thin stand that may not be economical to take to harvest. So what are we doing here to try to understand winter hardiness? Well, one variable that we're interested in is genetics. What varieties of winter barley might be more or less prone to those winter injury and winter kill issues. So in this study we have 3 varieties of winter barley that are known from previous research to be more winter hardy. And then we also have the Czech of the cereal rye that is known to be hardy in this climate. And then we have 4 winter barley varieties in here that are known to be less winter hardy, that we would expect to not do as well at our northern locations. There's 4 replicates of those 7 barley varieties and 1 rye variety out here. And you can see visually right away where the rye is. The green plots are the cereal rye, the Czech in this case, and the more white, not so happy, more spotty looking plots are the winter barley. As you look out across this, you can see that some have more green and some have more brown. So already there's some visual signs here that some of these varieties fared better than others. And I just walked through before we started filming today and I noted the ones that have more green. The one variety that seems to have more green out here is Malt. Malt is a pretty widely known winter barley variety. Funny enough, it is one of the varieties that we had categorized as poor winter hardiness that actually seems to have done better in this particular circumstance. So a lot of times in research we set out with one idea and end up coming away with another. So it may prove that Malt actually should be in that better winter hardiness category relative to some of the other genetics that we have here. So genetics are one component that we're interested in with this study. The other component is environment. So this is what we call a G by E study in agriculture, genetics by environment. We're also doing a lot of on-the-ground environmental monitoring in this study to look at specifically what parameters might be important and what the thresholds of these different parameters could be. So I'm standing by these stakes that contain some of our monitoring equipment. The white here is a snow depth stake, so this is just a stake with demarcations of inches and feet on it. And then we have a trail camera that you would use for scouting deer or any other type of outdoor activity like that. And we have that trail camera set up to take 3 images a day at 11 a.m., noon, and 1 p.m. And what that does is every day that gives us 3 opportunities to take a picture of this snow stake and gauge snow depth based on that stake. And every replication here has a set of sensors and monitoring tools here. Aside from directly measuring snow depth with the stakes, we're also using an indirect method of measuring snow depth with one of these sensors down here. It's a hobo sensor that measures both temperature and also light intensity. So we place the sensor at the crop level, and what we're able to see is as the snow accumulates, the light reaching that sensor decreases. And so it's an indirect way of measuring snow depth, and we will be able to look at the correlation between that and our direct measure of snow depth with the cameras and see what kind of relationship there is there. That same hobo sensor is measuring temperature, and so we can measure that temperature regularly, and we have that data set accumulated. These plots were planted on September 10th last year, and we've continuously monitored that with those sensors logging that data all the way through. The other sensor that we have on the back side of this stake is a temperature and humidity sensor. It's a relative humidity sensor. Probably would be better to actually measure soil moisture or plant tissue moisture in some way, but this is the best way we can come up with to remotely monitor over the winter at a very reasonable cost. So we're going to be able to look at this spring which of these varieties survived and to what extent, and the relationship between that survival and the genetics, the varieties, and then also the relationship between that survival information and the environmental data that we have from these sensors. You can see that we have signs of a lot of winter injury in these plots here at Chatham. Conversely, We've seen pictures from Empire and from KBS, and they have a lot of green looking barley. At KBS, it's already greening up and growing and looking quite healthy. So we have a good gradient across locations this year in terms of looking at the environmental differences that might be important for this crop and where that environmental threshold or climatic threshold or maybe a latitude line that we can draw in the sand as far as where we can grow this crop successfully. So we're inside, so I can show you a little bit more detail on a little bit about the environmental components of our locations and also some of the monitoring that we're doing for this winter barley hardiness project. One of the challenges actually in Michigan with a crop like this and the issues of winter hardiness is the fact that our microclimates and our hardiness zones are not straight latitude. We have the lake effect, and the lake effect creates a sort of target or bullseye pattern in Michigan, and particularly in the lower peninsula, where nearness to the lake may be as important as latitude for extreme temperatures. This is some of the data from one of the hobo sensors in our field. So this is Rep 4 here at Chatham. And this is monitoring all the way from planting back on September 10th, all the way till just the other day when I downloaded the data just to check the sensors. What we're looking at here is both temperature and the light intensity. The temperature is the black, and you can see here we started in the 70s, mid-60s. When we planted in September, that quickly declined. We had a bit of a warm-up in November. And then temperatures kind of came down around 30 degrees and actually held there. However, when you look at the light intensity as a sort of proxy for snow depth, you can see that we had a lot of our winter without significant snow cover, the early part of the winter. Our first significant snow came at the end of October, and you can see the little dip here in the light that indicated our first snow cover. But we lost that, and there wasn't a lot more snow cover for quite some time until we really got into January, where we had significant snow, and you can see the light nearly disappeared as that snow accumulated. Snow is insulating, has an insulation factor, and so snow cover is thought to be good for survival in these overwintering small grains. And it is interesting, when we had snow cover, temperatures at the ground level where the crop was or staying right around freezing or maybe just a little bit above. Then when our snow melted, the temperatures actually dropped pretty dramatically. Early March, you can see we had a cold night with no snow, and our temps quickly dropped below 20 degrees, whereas under the snow they had mostly stayed around freezing or just a little bit above and below. How are we really measuring how the crop responds to these environmental factors? In the fall, we took stand counts, and then we're going to repeat that around this time in the spring at green up, as soon as we can tell the difference between a live plant and a dead plant. The other thing we're going to try to do this spring is use an application called Canopio. When we get to sort of full tiller or just before stem elongation at Feeks 4 or 5 growth stage, we're going to use this app that takes a photo of the plot and can discriminate green from other colors and give us a % green area or canopy cover, a way of addition to the number of plants there, how vigorous, how large they are, how many leaves or tillers they might have as measured by % green area or canopy cover. There's also a physiological component to this project, which is pretty interesting. We're trying to not only look at how this G by E interaction occurs, but try to understand some of the mechanisms behind winter hardiness in barley as well. A lot of what determines what we see in the spring now, this time of year, is actually what happened last fall. And that is what is called the hardening process itself. And that is the transition that the crop goes through physiologically to adapt to colder temperatures as it goes through the fall and into the winter. There's some things that are happening in that process that we're trying to understand more about, and Dr. Nikhil Jaikumar is the lead on this portion, along with Emily Merowitz-Holm and her graduate student, to analyze some tissue samples from these plots to get at some of these mechanisms. And one is oxidative stress. Antioxidants help deal with oxidation in living things, and plants deal with the same issue of this oxidative stress. And we're interested in understanding how that might contribute to winter hardiness and winter survival, and the ability of these different plants to deal with that. The other thing is DNA repair. So these stressful environmental conditions can actually damage plant DNA. These plants have mechanisms to try to repair that DNA so that when they're making new cells as they grow and develop, that they're doing so as intended to complete the life cycle of that crop. So we took tissue samples at 2 times in the fall, once before significant freezing weather and once after significant freezing weather, to look at how enzyme activity and DNA repair, dealing with oxidative stress and this DNA degradation that can happen due to these environmental conditions, and to try to understand how winter barley as a species is reacting to these stressful conditions, and then if there may be differences in varieties. That's especially important where that Czech rye comes in, because rye is, at least a lot of varieties of rye are very successful in terms of surviving cold conditions and stress that comes with winter time. And so we can do some relative comparisons between all our winter barley samples and the rye as well. We will develop a report for our funders at the Craft Beverage Council and to share with others. The nice thing about this project is that it has an applied component and it has a more theoretical component. I think what we just talked about, sort of the mechanisms and the laboratory portion, are very kind of theoretical and science driven, and we'll probably have some publications, some peer-reviewed journal publications that come out of that effort in particular. But the applied knowledge is more for the grower to say, can I grow winter barley in my environment? And if so, which varieties are more likely to be successful? The physiology portion is more oriented at breeders, like Kevin Smith at Minnesota, who's breeding winter barley and needs to know what traits or what mechanisms do I target or select for as I'm making those selections, so that hopefully in the future there's a lot more varieties of winter barley that can survive an environment like this, and we're not having this same issue of risk for the grower wondering, is barley going to make it this year or not. Even at southern locations like Kellogg Biological Station or campus, or in northern Indiana or Ohio, you have had years where winter barley does great, and you have years where winter barley is a total loss. So it's likely not just a latitudinal threshold. It's much more about microclimates, and it's much more about ability of these varieties in this crop to adapt given the conditions, and the variability that will only be exacerbated by climate change, and why we're thinking about winter barley as an adaptation tool.
That's what's up at UpRec. Hope to see you again next time.
Anything you want, we got it right here in the old UP.